Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Opportunities and Considerations for Inhaled Biologics. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Kindeva Drug Delivery, who developed the content for this presentation. Kindeva Drug Delivery is a global contract development and manufacturing organization focused on drug-device combination products. They develop and manufacture products across a broad range of drug delivery formats, including pulmonary and nasal injectable and transdermal. Their services offerings span early-stage feasibility through commercial-scale drug product fill finish, container closure system manufacturing, and drug device product assembly. Kindeva serves a global client base from their state-of-the-art manufacturing research and development facilities located across the U.S. and UK. Now it's my pleasure to introduce our speakers for today's webinar. And first, I'd like to introduce you to Dr. John Patton. There he is, head of Kindeva Scientific Advisory Board, Kindeva Drug Delivery. John is a drug delivery scientist and entrepreneur with extensive experience in biotech and the development of new inhaled medications. He serves as executive chairman, co-chairman, director, and advisory board member for multiple companies. John has more than 110 publications. He is an inventor or co-inventor of more than 50 patents. He is also the 2024 winner of the Charles G. Thiel Award for Outstanding Research and Discovery in the Respiratory Drug Delivery at the Respiratory Drug Delivery Conference. And next we have Dr. Keith Ung, Director and Bay Area Site Head. There he is, drug delivery with over 3 decades of industry experience. Keith is an expert on inhalation, infrastructure setup, and characterization of pulmonary and nasal drug delivery system. In his role at Kindeva Drug Delivery, he oversees the Bay Area facility, leveraging extensive expertise in the vitro characterization of spray, dried, peptide, and small molecule powders and pulmonary delivery systems in support of proof of concept, preclinical, and clinical studies. Keith was previously the CEO, president, director, and founder of iPharma Labs, Inc. iPharma was acquired by Kindeva Drug Delivery in 2022. And next is Dr. Andy Clark, President and General Manager at Aerogen Pharma Corporation. Hi there, Andy. Andy is a seasoned pharmaceutical executive with over 35 years of experience in respiratory drug development. His experience includes managing the development of some of the pharmaceuticals industry's most novel inhalation products. Over his career, Andy has received the Young Investigator Award, the Thomas T. Mercer Award, and the 2012 winner of the Charles G. Thiel Award for Outstanding Research and Discovery in Respiratory Drug Delivery at the RDD Conference. And next, it's my pleasure to introduce you to Dr. Jeff Weirs, Chief Technology Officer at Systemic Medicines, Inc. Jeff is a physical chemist with a broad disciplinary and scientific background and 30 years of experience in colloid-based research and development. Jeff has published more than 100 peer-reviewed articles and is a prolific inventor with more than 70 issued U.S. patents. His inventions have contributed to 6 approved respiratory products. He is also the Charles winner of the Charles G. Thiel Award for Outstanding Research and Discovery in Respiratory Drug Delivery at the Respiratory Drug Delivery Conference. So there you go, our speakers. And now it's my pleasure to pass the mic over to John. So John, when you're ready, you may begin.
Thank you, Sonia. Good morning. We're all here in San Francisco Bay Area, but I know you're scattered all over the world, probably, we hope. Thank you for coming. We're excited to tell you 4 little stories here. If I can have the next slide. So biologics are about 10% of all drugs being inhaled, considered for an inhalation. Um, they are the sexy drugs of today. Everybody wants to be doing biologics. The small molecules, which are fantastic drugs, of course, have kind of taken a little bit of a backseat to the biologics, but there's a number of issues with biologics, and we'll talk about those in just a second. Um, the delivery system that you choose for your biologic is kind of dependent on the molecule and the disease and the market and the cost and all kinds of things. So there's not one size fits all. Each application really requires different formulation, different device. One of the really huge things that's been happening in inhalation science are engineered fine powders, and you're going to hear some really great talks here in just a second on those powders and how special they are. But we still have nebulizers, and nebulizers have a very strong place in inhalation drug delivery. They're very good for the elderly and children. They're very good for getting quick clinical results, proof of concept before you get into any really commercial product. You can do a quick nebulizer trial. And then the other thing that's really happening in medicine today is we don't want to be giving drugs to people every day if we can prevent them. And vaccine science is really exploding. There's lots of vaccines for infection, COVID. We all know about that, but they're making vaccines now for asthma, for Alzheimer's, for cancer. So much better to have a vaccine given a couple times maybe, and then you're good to go. Your disease essentially is not present. Next slide, please, Keith. So what is a biologic? I mean, in some ways, the best definition is it's not a small molecule, because lots of small molecules are biological. I mean, look, there's glucose on the list. We really characterize biologics as molecules that are bigger than 1,000 Daltons, and 1 Dalton is a hydrogen atom. So 75% of all drugs are these small molecules. But then as you get up over 1,000 Daltons, you get into the biologics. And biologics are basically polymers of amino acids, polymers of nucleic acids, and a lot of the proteins actually have polymers of carbohydrates. It's called glycosylation. So these molecules are far more sophisticated and larger than the small molecules. And you can look at, as you go down the list here, the flu virus, giant number of Daltons, a bacterium, trillions of Daltons, and I don't even know what that number is for a stem cell. So the biologics are big molecules. Small molecules are considered to be promiscuous in the sense that because of their small size, they can sometimes get in trouble and have some toxicities that you might not get with a big molecule. But that doesn't mean that biologics don't have their safety issues too. They do. The other thing is that the small molecules are regulated by one branch of the FDA, CDER, and then the biologics are regulated by a different branch of the FDA called CBER. Next slide, please. So if we look at the pulmonary biological pipeline, Bars and Gavtash at Kendeva has put this data together for us, and it's from the database PharmaCircle. 227 programs of pulmonary biologics are ongoing right now, and you can see the list on the side. Very diverse, but roughly 50% are polypeptides, amino acid based, proteins, peptides, antibodies, and then the other half are nucleic acid based. Things like mRNA, the COVID vaccines were mRNA, gene delivery. So there's about a 50-50 break here. But there's also, and this is for inhalation through our mouth down into our lungs. But there's also a very large number of nasal biologics in development. About 37% of those are vaccines, and only about 5% of the pulmonary ones are vaccines right now. But look at the variety there. So next slide, Keith. So there you see the stage of development of all these really innovative new therapeutics. The amino acid based, there's only a handful of approved biologics for inhalation. There's an enzyme, DNase for cystic fibrosis, and there's a couple of forms of insulin, but there's just a few. But look at all these in development, and a lot of them are in early stage development, preclinical. And this does not include all the programs in universities. They have been excluded from this representation. So with that sort of introduction of very complex kinds of therapeutics in the biologic space, I think let's see the next slide. So now we're going to go into these short pieces, and we're going to start with Andy talking about nebulized proteins. That's water-based nebulizers and lung surfactant to premature babies. So I'll turn that over to you, Andy.
Yeah, good morning, everybody. Like John said, we're in California, so it's morning in California, but I guess good day is the Australian way of saying it, and it's time independent. So I'm going to talk a few minutes about nebulized proteins, and I'm going to do two things. One is just generally talk about nebulized proteins and peptides, and then specifically about a product that I guess I've spent the last 8 years of my life helping develop, i.e. aerosolized lung surfactant. But before we get into that, I thought it'd be interesting just to put nebulizers in their place in the hierarchy and the grand universe of inhalers. This is a diagram that I think has been evolving over the years between Jeff Weirs and I. And the line to look at are the images of the different inhalers, and the line underneath that says maximum lung dose per actuation or per puff. And what you can see is at the bottom end, you've got the MDIs and the smart mist inhalers. Limited in the volume that they can atomize, and hence limited in the amount of drug they can actually deliver per actuation. And then at the top end, you've got dry powder inhalers and nebulizers. And there's a reason this talk is focusing on nebulizers and dry powder inhalers. And that's because most of the proteins and peptides that will be delivered via the pulmonary route are fairly large doses. And of course, the powders and the nebulizers are quite capable of those large doses, whereas the smaller volumes and dose capabilities of the other two dosage forms are really limited in terms of what you can do with them. So if you can go to the next slide, Keith. So let's get started with the two main types of nebulizers that are available these days. The one on the left are the jet nebulizers, the one on the right are the mesh nebulizers. And the jet nebulizers, from a protein perspective, really have three problems. The first one is all to do with atomization. And when the solution that's placed in the nebulizer is atomized, it actually makes quite a coarse droplet size distribution. And the majority of those droplets are captured and put back into the reservoir and go through the atomizer time and time and time again. So one of the problems is the proteins that are in solution get exposed to those shear forces many, many times before they get out of the nebulizer as droplets in the right size range to be inhaled. So one thing you have to be very careful of is the stability of the molecules to those shear forces. The second one is the dead volume, and this is all about efficiency. Generally, because of the way these nebulizers work, they have a fairly large surface area internally, and drug gets captured on those surfaces. And of course, that reduces the efficiency. And again, from a protein perspective, very expensive molecules, you don't want to waste half a mil or a mil of that solution every time you try to deliver a dose. So really the major ones are shear stress and efficiency. And nebulized proteins, peptides, the majority of them would survive the mesh nebulizers on the right, but the problem with the one on the left is some of them do, some of them don't. So for example, I've got pulmozyme there. That's a molecule I worked on back in the early 90s. Pulmozyme is absolutely fine in jet nebulizers, doesn't do any harm, comes out intact without any aggregation or degradation. On the bottom, I've got human growth hormone. And human growth hormone is essentially problematic as a nebulized solution, full stop. Growth hormone tends to not like the air-liquid interface in droplets and unfolds. And when it unfolds, it aggregates, so you end up generating aggregates when you're trying to deliver it via an aerosol. So if we move to the right-hand side, we've got vibrating mesh nebulizers. They have a couple of advantages over jets. One is the molecule only makes one pass through the hole. So the mesh nebulizers are little holes that are vibrated, and every time you vibrate the hole, as you can see from the movie, you generate a droplet. Reality is you generate a few droplets, but the movie is reasonably okay in terms of trying to show what's going on. So the solution only goes through the hole once, and as a result, the shear forces that the molecules are exposed to are a lot less than the jet nebulizers. The other kind of major advantage is really efficiency. Because you're not trying to capture and recirculate, you don't have large surface areas, so they're much more efficient in terms of the fraction of the fraction of the original nominal dose you put in the nebulizer that gets out to the patient. So can I have the next slide? So this is just a list, and this is courtesy of a colleague of mine, Roland McLaughlin, over at Aerogen Limited in Galway. And we put this list together, and they're the molecules that we know are being investigated as nebulized solutions. And you can see they range all the way from insulin at 6 kilodaltons all the way up to antibodies at 150-ish kilodaltons. And then finally, the one at the bottom is our Aerofac formulation, which is a bovine-derived lung surfactant, which has got lipids, SPB, and SPC surfactants in it. So a very wide range, and the reason these molecules are on this list is because they all survive the nebulization process intact, and so are amenable to delivery via this route. So I can have the next slide, Keith. So this one is a little bit about the rules, and somewhat the rules of aerosol delivery in general, and somewhat the rules of aerosol delivery specifically by nebulizers. What I've got on the left-hand side is a very old plot from a paper back in the mid-90s looking at the regional deposition of aerosol delivered via nebulizers. And unfortunately, I mean, these data are all from scintigraphy studies. And unfortunately, with scintigraphy studies, all you get to do is measure what's in the head and what's in the lungs. You generally don't get data that tells you how much of the delivered dose actually deposited in the body. So this plot is really regional deposition rather than total lung deposition. And you can see, not surprisingly, as the droplet size gets smaller, you put less in the head and you put more in the lungs. And the typical range here for most nebulizers in this 3 to 5 to 6 sort of microns, where you get a split between what deposits in the head and what deposits in the lung, somewhere around 80% in the lung at the smaller size and about 60% in the lung at the larger size. So that gives you some clue about droplet size requirements. And then on the right-hand side, we've got lung deposition as a function of aerodynamic size. And this is a true lung deposition. It's not regional deposition. This is how much of the inhaled dose actually deposits in the lungs and avoids the head or avoids being exhaled. And the point I want to make to this plot is there's always a trade-off. And this is particularly with nebulizers and some work we did with preterm infants. But the principles apply to adults and to children too. And you can see that the average, which is the open square boxes in the middle, that's the average deposition you would get across a population. And you can see where the error is for about, 4 microns, that's somewhere around 35%. Now, the arrows show the variability. The upper line, the open diamonds, and the lower line, the red circles, actually show the 95% confidence limits across a population. And you can see that at the maximum mean deposition, you've got an awful lot of variability. The best patient gets somewhere around 50% in the lungs, the worst patient somewhere around 12% in the lungs. And if you want to avoid that variability, you have to make the aerosol smaller, which avoids deposition in the head. But of course, you can get the variability down, but then you're losing out a little bit on the mean deposition. So you can see there the mean deposition is about 25. If you're down around 2 microns, the maximum is about 28, the minimum is about 18 or so percent. So what I'm really trying to illustrate here is you're trying to hit a balance between the maximum deposition on average and the variability between patients. And obviously that's really important, be it a general population you're treating or a clinical trial design you're trying to do. So if you want to move on to the next slide, Keith. So that's some general principles about nebulization of proteins and about aerosol deposition. I've got a couple of slides here just to talk about the product we've been developing for the last few years, and that's aerosolized surfactant to be delivered to preterm infants. And why would we be interested in doing that? Well, the photograph here on the right-hand side in the middle is actually a photograph of a procedure of how lung surfactant is delivered to these very small babies as of now. Lung surfactant is an amazing, I was going to say molecule, but it's an amazing drug product. When it was first discovered in the late 80s, early 90s, it actually more than halved the mortality rate for preterm infants. Preterm being anywhere between around 24, 26 weeks, all the way up to 32, 34 weeks. So surfactant's really good. The problem is, as our CMO always says, the delivery procedure is the crowbar and the flashlight. The crowbar is the laryngoscope that's in the neonatologist's hands here. And they usually shine a torch into the mouth or a flashlight into the mouth so they can see where they're actually pushing that blue tube, which they put into the trachea, and then they inject the lung surfactant solution directly into the lungs. Then they have to put a bag on the baby to make sure the surfactant disperses. And quite often they have to sedate these babies, which is really bad news for very early premature infants, because they'll fight this process, not surprisingly. So that's why inhaled surfactant or aerosolized surfactant is such a holy grail in neonatology. But from an aerosol perspective, it's problematic. If you look at the plot on or the picture on the left, these babies breathe extremely rapidly. They breathe with very small lung volumes, and they really have very tiny airways. So there's a real technical challenge in terms of trying to get aerosolized surfactant in through the nose of these babies, because they're obligatory nose breathers, and down through their nasopharynx and actually into the airways. So can I have the next slide, Keith? So this is how you meet the technical challenge, and it's really a triad. The first one is efficient delivery, and that diagram is actually a diagram of the interface that we use to generate the aerosol that's going to be inhaled by the baby. And the reason efficiency is important is because lung surfactants are expensive. Usually per animal, when the lung surfactant is actually being extracted, one animal will make about 4 doses. So they're quite expensive in terms of both animals and in terms of cost. So the first one is that efficient delivery. The second one is droplet size. We already talked a little bit about that, and the challenge there is making sure you've got that balance between variability between neonates, because they're no different to adults. They have very variable geometry in their nasopharyngeal regions. So droplet size is important in terms of a choice and getting that balance between variability and absolute mean deposition. And then finally, these babies have an inspiration to expiration ratio, typically 1 to 2. So if you develop or deliver aerosol continuously, you're throwing away 2/3 of the aerosol during expiration. So you have to be able to breath sink. And further from that, you have to be able to breath sink in the first part of the baby's breath. You don't want to be putting aerosol in at the end of a breath, because it goes into the dead space in the airways. That won't deposit, and essentially it almost immediately gets exhaled. So quite a few technical challenges to get right. The picture on the right-hand side here is actually, that's a mannequin rather than a preterm infant, but that's the system actually on the baby. And the bigger picture is a picture of the controller that actually takes note of the baby's breathing frequency and delivers aerosol during inspiration only, of the correct size from the PDAP mesh technology that generates droplets. a little bit smaller than your average mesh nebulizer. So this system has actually gone through a phase 2A study in Australia. It's gone through a phase 2B study in North America, and phase 3 is pending, as they like to say in this industry. So if you can go to the next slide, Keith. So finally, just a quick summary. With some exceptions, most proteins are stable to nebulization, particularly by mesh nebulizers, because their lack of shear. Mesh nebulizers being, extremely efficient. Growth hormones probably one of the exceptions. Any molecule that doesn't like air-liquid interfaces is going to be problematic with the nebulizer and probably has to go the dry powder route. Nebulizers are not necessarily the best modality in terms of an end product, but they're certainly, as John said earlier on, fast. And pulmozyme is a really good example. Pulmozyme, I was involved in the latter half of the development, and that was less than 5 years from cloning on a bench to actually get an FDA approval. Aerosolized surfactant looks like it's going to get real, and we're going to actually find the holy grail. And like I said on the previous slide, we've got through 2A and 2B so far, and phase 3 is pending. And with that, I'm going to hand it over to Jeff.
Thank you, Andy. Next slide, please. So spray drying is a mature technology for creation of powders for inhalation. It's a single-step process, but we can subdivide it into sort of 4 sub-processes. Making a feedstock, which in the case of proteins, is a solution that's dissolved, a solution of the protein dissolved in water or ethanol water mixtures. We then atomize those droplets to create 8 to 10 micron droplets that are then dried in a drying chamber. And ultimately, we collect those in a cyclone or a bag house in terms of separation from the airstream. And that's the process. One of the key things is that there are a number of physical stresses that can occur on the proteins during the spray drying process, and you can see them listed there. Temperature, increases in shear during the atomization event, adsorption at the air-water interface, and potential issues with dehydration of the protein. Next slide, please. So what we're going to do here is we're going to spend some time talking about how to mitigate those issues. So first of all, let's talk a little bit about the thermal history of the droplet and the particle. And this is courtesy of a slide from Herm Snyder and David Lechuga. And so what you have here is on the y-axis there is the calculated droplet temperature as a function of time. The first thing that you'll note here is that once we atomize the droplet goes through what we call the constant rate evaporation period, where the temperature of the droplet and the protein that's dissolved within that droplet never really reaches the inlet temperature on the dryer, but rather is basically fixed there at what we call the wet bulb temperature, and that's due to evaporative cooling, which maintains that droplet temperature just above room temperature. The other thing to note there is that evaporation process is complete within just a few milliseconds. So the inlet temperature on the dryer, although it can be 120, 130 degrees, somewhere in that neighborhood, really the protein never sees those sorts of temperatures. But when you get to the point where you've now formed a particle, then the temperature of the particles can begin to increase. And that's it's consistent with what the outlet temperature on the dryer is as opposed to the inlet temperature, or for most cases, it's the collector temperature, which we can jack it and bring down some amount lower than the outlet temperature. The time scale for that process is what's important, whereas it's milliseconds for that initial evaporative piece. You can actually spend time in the collector for an hour or two. So that's really the rate determining or most important process parameter in terms of the impact of temperature on these proteins. Next slide. And so you can see here some data from Exubera. The panel on the left there, what you're looking at is a plot of the formation of high molecular weight proteins as a function of the outlet temperature on the dryer. Typically, the unfolding temperature for most dried proteins is greater than 100 degrees Celsius. And what you can see for Exubera there, inhaled insulin, is that when you get above about 120 degrees, you see a rapid increase in the formation of high molecular weight proteins within the formulation. So obviously, you want to maintain your outlet temperature well below that unfolding temperature for the protein. The panel on the right shows what happens, in terms of prolonged storage, depending upon the outlet temperature or the protein might see. And as you can see, again, consistent with the panel on the left, that you will, in essence, if you maintain the outlet temperature less than about 90 degrees or so, that you can maintain the stability of the protein quite nicely. Now, click again there, Keith. What you can see in the upper left-hand corner there is that's the time scale of the actual process, where we're at these elevated temperatures during a spray drying process, and so we don't anticipate significant degradation over the time period that we actually maintain at the outlet temperature or collector temperature. Next slide. So that was Exubera. At Inhale, we sort of progressed beyond that, and went into what we called our Pulmosol formulation technology. And the Pulmosol technology was associated with the creation of core shell particles, where we have a shell-forming excipient that basically is in charge of the micromeridic properties of the powder and its aerosol performance, and also in getting the protein off the interface. And that's usually something like leucine or trileucine and phospholipids. And the core of the particle, where we want the protein to be off of the interface, and we stabilize the protein within the core of the particle with glass-forming agents, buffers, antioxidants, whatever the protein needs in terms of maintaining its stability. One of the keys here is that we can create these core shell particles by controlling what's called the Peclet number, which is essentially a ratio of the diffusion within there to the evaporation, the rate of evaporation. So we can essentially, by controlling the composition of the feedstock and the drying event, we can basically push the shell forming agent onto the interface. Next slide. So what does that translate to? Here's some data from 3 different antibodies, and what you can see, they vary in molecular weight from about 55 kilodaltons up to 145, so a full antibody and then an antibody fragment. And essentially, they all use the same formulation, if you will, in terms of what we call our platform formulation. And you can see on the panel on the left-hand side there that the primary particle size distribution is really controlled by the shell-forming excipient and the drying conditions. And if we use the same drying conditions, independent of whether you have a drug loading of 25 to 38%, independent of the nature of the antibody, you get the same primary particle size distribution. So if you look at the table there on the right, what you can see is that the mass median diameter of the particles there is on the order of about 2 microns. The tap density is about 0.15 grams per cubic centimeter. The water content is low, so you can prevent plasticization of the protein. And then the aerosol properties on the right-hand side there, where you're looking at an emitted dose on the order of about 95%, and then MMAD, mass median aerodynamic diameter, that's essentially about 2 microns, which is optimal for maximizing the delivery into the lungs, and the fine particle fraction less than 3.3 microns, which is essentially a marker of more peripheral delivery within the lungs, expressed as a percentage of the nominal dose, is on the order of about 60%. that equates to about, in terms of fine particle fractions that we're used to seeing, less than 5 microns as a function of the emitted dose of about 80%. Next slide, please. So we can take that a step further. We can actually adjust the process in such a way to really try to bypass deposition entirely in the mouth and throat. And what you can see there on the left-hand side is a plot of the total lung dose as a function of aerodynamic diameter, and what we're achieving there for 2 smaller proteins and essentially a full or an antibody fragment there, CSJ117, which is a TSLP essentially antibody, is somewhere between 95 and 98% total lung doses measured with an Alberta throat. So the whole strategy behind what we did there was that we wanted to minimize the size of the primary particles from an aerodynamic perspective. And so we defined this parameter called dA, which is simply the mass median diameter of the primary particles that you measure by laser diffraction times the square root of the tap density. And if we keep that, the dA less than about 0.5 to 0.7, again, you can effectively bypass deposition in the mouth and throat. One other thing that's worth pointing out is that while insulin and relaxin, the 2 smaller proteins, form corrugated particles without any shell-forming excipient, the larger protein does not. And so the presence of the shell-forming excipient is critical to getting that corrugated morphology. And as you can see in the panel on the left, smooth particles have much lower total lung doses than do the corrugated particles. Next slide, please. So I want to finish here with just basically talking about the core of the particle now and what we're trying to do there. So this is data for a small protein called relaxin. And the black bars there represent essentially the intravenous formulation that made it through phase 3 clinical development, and both FDA and EMA basically bought off on the formulation and its stability. And that's at 5 degrees, so that's at refrigerated temperatures. Then you can see the advantage of what proteins that are made into a dry powder can bring from a stability perspective. So if you look at the red bar, that's just simply taking the concentrate from the intravenous formulation, which is just simply the protein in acetate buffer, and spray drying that. So it's a very simple formulation of just protein and buffer. And you get and that's stored at 25 degrees now instead of refrigerated temperature. So room temperature storage, and you see a benefit in terms of reducing oligomer formation and reducing essentially the formation of the isoasp. Finally, if we focus on what we called our platform formulation that we showed you, some of the ability to create the same types of particles independent of the nature of the protein that we put in it. Now, if you look at the oligomer formation by SEC, and this is after 6 months of storage at room temperature, you essentially see no increase in the oligomer formation, and hopefully that will result in reduced immunogenicity for the protein, since a lot of people think that there's a relationship between formation of high molecular weight proteins and immunogenicity. I want to finish by just commenting on packaging and how important it is. It's one thing that's not talked about a whole lot when we talk about inhaled proteins, but the packaging, to prevent essentially water ingress through the packaging, we really need tight packaging where we can, the shelf life of the product is going to be determined by the water ingress through that packaging. So it's really critical that we maintain a very good moisture barrier till the product is used. So with that, I will turn it over to Keith.
All right. So the question is, why spray drying? I think we've got to be agnostic about this. When we talk about taking a formulation, how do we have to put through the process? And I think we're reflecting back to Andy's chart about selecting what the litter platform and about what the dose size, really that when it comes down to the important part is that what why we're choosing spray drying. The main focus here is that this is a bacteria that is very sensitive to moisture. You have high moisture contact in that product, it can change the potency, you can change the dose over time. So the important part is just to reduce the amount of moisture in that product itself. The other one is that, with going back to that chart, is really the dose side. With this, so we're attempting to deliver millions of bacteria in a single dose. And that means that translates to higher payload, which the dose size is going to represent milligram scale. So it only fits in the DPI as in the liquid. Again, you should use water, and that is also be impractical just from the stability standpoint. The other one is nice thing about the spray drying, as Jeff mentioned, is that we can control how we want particles to look like. We can change its density to enhance its dispersibility. We can also, increase the API loading to get the right dose to fit into the cavity or the capsules that we intend to deliver. The other benefit with the spray drying is that we also demonstrate that over time is that you can increase the efficiency by changing the density of the particle, changing the skin formation of the particle. This would enhance the dispersibility and flowability of the particles. And John and Jeff mentioned in previous talks, if you make the powder flowable, low density, very dispersible, selecting the device to go with the powder can be very simple. It can be a two-piece part with a single straw, behave like an aerosol engine to get that. So in this case, this is an example where you have an indicator. Oops. You have indicator. Apology. You have indicator basically delivering two format of dry powder. One is a micronized, one is spray dry. So with the spray dry, we can definitely enhance the delivery efficiency by 2.5x delivery efficiency. Another part, what Jeff mentioned in his talk, is that protein peptide biologics can be very expensive. So having a high delivery efficiency is important. Some of the cost of the drug itself can be thousands of dollars per dose. In comparing to small molecule, there's $10, $15 for asthma COPD product. How do we make these? How do we spray dry? How do we prepare? This is basically how we start with the process. This is a simple. The plot to the right is the spray dry that we have. Everything is made of stainless steel. How we start with the process that we take it through the API, we disperse it in water. We basically determine that basically it's 25% loading is optimal for this formulation. We added the glass stabilized excipient. Again, the purpose of this is to protect the powder during the drying condition when exposed to temperature. The shell former is an important part. We also need to consider that in the formulation. In this case, we chose a shell former to enhance the dispersibility and flowability of the particles. And that's including other excipient. We started out with basically spray drying using a single atomizer nozzle. We spray dry, collecting the cyclone. And as you can see, is that with that we can produce 2 to 5 gram scale with a greater than 6% yield. We can make up to 20 to 50 gram scale in a single batch. In this case, we can increase the yield by more than 70%. In terms of the results, what we have with this particle, what we got is that post the spray dry product, we get 80% about viability of the drug product. We didn't do the microbe assay. The customer did the work in there, but demonstrate that we are getting more than 80%. What contained the particle is a live bacteria with amorphous excipients. And as you can see, the panel on the middle is basically the particle contains semi-quartic particle. The graph on the below essentially measure with laser diffraction 1.5 micron, mean geometric diameter of the particle. And the important part is the panel to the right was basically showing the isotherm plot of the moisture uptake of the particle. It tells us what it gives us the information is that when you encapsulate the powder, is that what condition should you be keeping the moisture level at, and also what it addresses that is important about the packaging configuration you need to consider when you actually package the drug product itself. And what we have here is basically is we're targeting a density somewhere less than 0.3, and that's just what we intended to create a very low density dispersible powder. Lastly, what we have here is putting the product into the device. We chose RS01 high flow resistant device, a size 3 capsule. We're able to fit 30 meg of powder into the capsule. Of that 30 meg, we deliver 25.6 meg as represented the emitted dose. Of that dose, it was measured about 5.4, 10 to the 7th colony forming unit in terms of dose, what we expect the patient to receive. The aerodynamic size for this powder with this device, we're getting about 2.3 micron. Geometric standard deviation is 1.7. This represents that we're basically creating a mono distribution aerosol. Of that, how much is actually represent the lung, we define that as the particle size less than 5 micron. Of the 30 meg of powder in the capsule, we're delivering 21.6 meg into the lungs. That represents about 72% of the nominal dose that it does with the starting capsule. The water activity is important as part of this product. As you know, in the food industry, is that we have to keep the water activity less than 0.1. We are measuring at 0.08, and it's important is in a sense that this product could be sitting in shelf life, it can be refrigerated. We don't want the dose to change over time, so it's important to maintain the low density. That means going back to Jeff's discussion about the important packaging, the consideration, the packaging, what that looks like, secondary packaging is required, include, just to minimize the moisture barrier. But in the spray drying process, we can definitely maintain the low moisture uptake, that over time, the packaging will determine the shelf life of the product itself. The coefficient, we measure about 1.5% of the moisture in the powder. The actual density that we measure as a bulk density, we got about 0.26 in this sense. So this campaign, when we actually start with this powder, we actually ran a total of 3 campaigns. And of that 3 campaign, we create at least 3 to 4 pounds of powder just to assess what are the metrics that we're looking for to develop the most optimized combination of the drug device that will go with to get a product performance out of what we want in that sense. So then in this case, I will pass it on to John.
Thanks a lot, Keith. Folks, Keith is also managing the slides for all of us, and he's also managing the pointer. And you're doing a fantastic job. Thank you so much. Okay, I'm going to finish up with real quick here because I see we're getting close on the time on 6 reasons why inhalation is better for a lot of vaccines. So there's 108 licensed vaccines through the FDA. 102 of them are by injection. For adults, it's mostly in the arm. And the 72% of the vaccine market is for pathogens that enter our bodies through our respiratory system. So we're sticking a vaccine in our muscle. It'll protect the lymph nodes. It'll really protect our shoulder from getting infected, but it doesn't do that as good a job as it could. Next slide, for protecting our lungs. So there's 3 arms of the immune system. There's the killer cells, the cellular immunity. These killer cells will take our cells that are infected and take them down and take them out of the system. And then humoral immunity. These are the antibodies that we build up against pathogens, circulate in the blood, ready to stick onto the microbes or the parasite, the virus or bacteria, which then signals for the immune system to attack them. And then there's a third very important immunity. It's called mucosal immunity, and this type of immunity it affects our secretions, the tears, the mucus in our respiratory system and gastrointestinal tract. And with this mucosal immunity, we are essentially pumping out a kind of antibody that's resistant to being broken down. It binds onto the microbes before they can get inside our body and helps us clear them. And look at the amount. It's 3.5 grams per day in an adult human can be produced. So this huge output in our secretions can be affected. Next slide. So if we look at the red in these figures of this woman represent the secretory immune response. You don't really get this very much if you get an injection in the arm. You don't get all those antibodies produced there. With the oral vaccines, you'll get some immunity secretory response in the head, and obviously in the gastrointestinal tract, and also in mom's breast milk. With the respiratory delivery, though, look at the coverage, and you know all of our inhalation systems do let some of the material go down to the gastrointestinal tract. So with respiratory inhalation, you have the potential to protect the lungs, the gastrointestinal system, the head, and the sexual organs. You can see that with rectal and vaginal vaccines locally given as suppositories, you get a very local secretory immune response, but it's substantial. So there's also another very interesting thing. Our skin, and Kindeva has two technologies. We have we have nasal and pulmonary delivery, but we also have micro needle products. And believe it or not, if you put a little patch on your skin, the tiny, tiny little points will go just inside the middle layer of your skin, the dermis, and there you also get protection of the lung. So the immune system is enormously complex and can do all kinds of protections if you put the vaccine in the right place. Next slide. So this is one of my heroes. George Washington got smallpox and survived. He was a huge fan of vaccination, which really came into America in around 1720. He insisted that Martha, his wife, get vaccinated, and he was militant, figuratively and really, in making his soldiers get vaccinated before they went into battle. The British were sending troops infected. It was one of the biggest factors in the Revolutionary War. It was keeping the soldiers healthy. And their vaccine was by scratching pox sore material on the arm into that dermal layer that I was just telling you about, and also inhaling the scratch material through the nose. And this protected Washington's troops. So here's something over 200 years ago where we knew that this kind of mucosal vaccination and dermal vaccination was effective. So next slide. Here's an example of a aerosol flu vaccine study back in 50 years ago, showing much better results with the inhaled than the injected, and significantly lower side effects. We all know from the pandemic, some of us got inoculations, and we got as sick as people who got sick on COVID. And it looks like with these mucosal vaccines, you can lower the dose substantially, increase the bioavailability, and really lower the side effects. Next slide. And then there's the fear of needles. I don't want to go into this. This is a meta-analysis of 35 studies. This does not go away. And guess what? These people who don't like needles, they're the sickest diabetics. And we all know people who did not take shots and passed away in the pandemic. So go to the next slide. So our goal at Kindeva is, and you saw these marvelous talks, Keith and Jeff on spray drying, that this technology has really been harnessed. And what we want to provide are these room temperature stable vaccine and other biologic products. You can see those little particles, 100 nanometer particles on the left are lipid nanoparticles, but they're also gene delivery particles, but they can be different sizes. But I just put the 100 nanometer one because it's so famous now in mRNA delivery and gene delivery. So last slide. The reasons we get a better immune response for pathogens that enter our lungs and our noses and mucosal systems, fewer side effects, and avoid needle hesitancy. And then you saw with these phenomenal talks on powder engineering and what you can do with those in Jeff and Keith's talks, and then maybe enable self-administration so you can get your vaccine in the mail, cut down on healthcare costs. And if you make your device correct, you can look at it, and if you didn't get enough on the first puff, you can take another puff to make sure you got it all in. So with that, I'm going to turn it over to Sonia, and thanks everybody. Let's go to Q&A.
Okay, well, thank you very much for that great presentation. I hope everyone enjoyed it. And with that, we'll put on our webcams, and I believe we have time just to squeeze in one question here. So just as a reminder to our audience members, you still have the opportunity to send in your questions, but remember the team will follow up with you after this presentation. So if I can just get all the other speakers to join us so everyone can see you one more time, and then we'll get into this question here. So this question is, this audience member is asking, what is the environmental impact of inhaled biologics, and how can their development best be integrated with sustainability initiatives like green propellants? Who would like to start off with that question, with that answer for that question?
Andy, why don't you take that
Andy?
one?
Thank you, Andy.
Yeah, so I guess one of my lead slides was the hierarchy of pulmonary delivery technologies. And reality is most of these proteins are fairly high dose, and most of them really probably aren't suitable for MDIs anyway. So, the great debate about replacing current propellants because of their global warming potential with less global warmers, less ozone depleting, is really probably not that applicable to most of these molecules. But where it is, it's obviously an issue of developing formulations with the new propellants rather than the old HFA propellants.
Sonia? Thanks, Andy. Sonia, can we go to the questions from the audience now?
We have not. Let's see if I can get one in here. Okay, so this question is for Jeff. Unfolding temps for monoclonal domains is typically between 65 and 80 degrees Celsius. Are no HMW species formed following drying due to exposure to the outlet temperature? Who would like to tackle that question, and then we'll close out the webinar?
That's you, Jeff.
That's you, Jeff.
Yeah, so I mean, it depends on the protein, obviously, that you're delivering to the conditions that you're going to choose on your spray dryer to. But yes, I mean, a critical process parameter is that you want to maintain your outlet temperature to be less than that unfolding temperature. So you have to adjust your conditions to be able to do that.
Okay, thank you so much, Jeff. Well, we've gone over time now, so I just want to say thank you very much, everyone, for sending in those questions. If we could not attend to your questions, the team will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see there on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. I'm going to share with you a link for the recording of this event, and you can share this link with your colleagues once they register for the recording here as well. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. So I encourage you to do that. Now please join us in thanking our speakers for their time here today. So thank you very much, John, Andy, Keith, and Jeff, for your time. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye, John, Jeff, Andy,
Thanks,
Keith.
guys.
Bye. Bye, everyone.
Thank you, everyone.